Okay, so prophase, just reviewing these major phases then. Prophase is characterized, so we have an animal cell shown on top and a plant cell on the bottom. This is from your textbook. So in both cases, we're going to see the nuclear membrane start to dissolve. So the nuclear membrane's breaking down and nucleolus disappears and the chromatin is condensing to form chromosomes. So that's a big factor in identifying as we're looking for chromosomes and we're looking for the nuclear membrane to break down. So that's prophase. By the end of prophase, we're going to start to see visible chromosomes. And the spindle is in the process of forming, but you're not going to see that in the microscope. So just look for visible chromosomes. You know you're in late prophase. So we're looking at those chromosomes showing up here and no nuclear membrane. So I can see chromosomes, but no membrane. It's really hard to write with this mouse. Okay, metaphase, that's when we said the spindle forms and pushes them to the center. So by the time we're in the end of metaphase, we should see chromosomes are lined up in the middle. And that's the key thing, is looking for chromosomes lined up in a line in the middle. So metaphase, middle. So I'm not going to show you, we're not going to hold you accountable for somewhere in early metaphase because there's things going on that haven't resulted in that final purpose of getting them lined up in the middle. So we're just going to have you look at it at the end of metaphase. So same thing with the plant cell. We start to see them lined up in the middle. Anaphase is when they're being pulled apart. So you're always going to look for a space between those chromosomes in the middle. We can see here there's a space between the chromosomes as those sister chromatids are pulled apart. So the sister chromatids are separated. We're pulling them apart, so one goes each direction. So here I can see there's a space as well, right? They're being pulled apart. And then telophase, again, we see a cleavage furrow only in animal cells. It's hard to see it in this slide. But here, this cleavage furrow kind of gives a figure 8 appearance. That is only in animal cells. Plant cells, we only have the cell plate, unique to plant cells. No, it doesn't work. All right. So that's what you're going to look for if you're looking for telophase in the microscope. You're going to look for a figure 8 appearance. You can see how that's a figure 8, and I can still see visible chromosomes. I don't see a nuclear membrane. I know where I'm at the end of mitosis. Same thing here. I can see visible chromosomes. I don't see a nuclear membrane. I see a real faint line, which is that cell plate. So the cleavage furrow is unique to animal cells. And then the cell plate is unique to plant cells. So what happens then? Sometimes we have mutations that have developed during DNA replication, or we have abnormal cell growth, again, resulting from mutation that is cancer, and the body can recognize abnormal cell growth and kill cells that have too many mutations. And we call cell death, which is programmed by the body and is controlled by molecules like tumor necrosis factor is something that we have in our blood that attacks little tumors as they grow. We're always growing little tumors, but we have factors in our immune system that attacks those cells and prevents them from growing. So some people, though, genetically, their family history, they just have a history of cancers. And that means that there's probably some dysfunction in the immune system that's preventing those factors in our immune function or immune system that prevents the growth of tumors. So there's something that's not functioning correctly. So we call that apoptosis. The P is silent, so it's apoptosis. So this is a long O, and this is silent. It looks like apoptosis, but it's not. It's called apoptosis, okay? And that's programmed cell death. That's the definition of apoptosis. And it's a good thing because we don't want cells growing out of control. So here's a cell, it's dividing, it's got problems, and it breaks down. So apoptosis is something that prevents cell growth and mitosis. So one protein that controls cell growth is P53, and it kicks in at G1. So where is G1? If you look at your cell cycle slide, I don't want to scroll back in the PowerPoint, but the cell cycle slide is on slide 194 in your notes. So G1, when does that happen? Before or after mitosis? It's the growth phase. So when do you think a cell would be growing? Before or after mitosis? Right after mitosis. So if something happened during mitosis and that DNA replication that led into mitosis, that will destroy that cell. And the signal protein, like I said, is P53. So it kicks in when DNA is damaged. And again, damaged DNA is just another term for a mutation. So we're not going to look at the details of that. Just know that it binds to damaged DNA, tries to repair the damage, and if it can't, it destroys the cell. So there's another term that you've heard related to cells, cell growth, mitosis, and that's stem cells. So stem cells are early cells that haven't divided to become specialized cells yet. So they have the DNA of that organism, but they're not specialized to be a heart cell or a brain cell or a skin cell yet. So they always have the ability to divide. So like once you, for example, once your brain develops, those cells don't divide anymore. We talked about that. Those are resistant to mitosis. You have all the brain cells you'll ever need when you're done with puberty. But stem cells can always divide. And we find stem cells even in adult humans. We find them in the bone marrow. We have cells that can divide into many types of different red blood cells or white blood cells. There's not many. There's only one type of red blood cell, but there's many immune cells, white blood cells. So they can become these cells. They're formed in the bone marrow, and then they mature in other parts of the body, like the thymus during childhood, or in the bone marrow. They stay there and mature. So we have these stem cells that we can use to grow into whatever we want them to grow into. And there's a stem cell research lab right now in Madison, like I said, and I've taken my biology students to that, and they're growing heart muscle cells, and that cluster of cells actually beats like a heart. We've got a long way to go, like I said, to turn it into a functioning organ, but we're doing a lot of research and learning a lot in this process. So cloning is taking stem cells and dividing them and making more and more stem cells, and then giving them the signals they need to become the specific cell types we want. So we're trying to help people that have genetic conditions like cystic fibrosis, which affects the lungs. We're trying to clone cells to repair and replace the dysfunctional cells that cause the lung disease in cystic fibrosis. Reproductive cloning is when we want to make a new individual. Have you heard of Dolly the sheep? Years ago, we cloned a sheep. I think we cloned a dog, trying to take embryonic stem cells. So you're taking stem cells from the early embryo and turning that into a brand new individual. And that person would be an exact copy of the original organism that we're taking those stem cells from. So what is the benefit of reproductive cloning? Well, think of it in terms of agriculture. What would be the benefit of cloning a plant? How do you think that could benefit agriculture and food production? In what way? Like, give me an example. Yeah, exactly. Genetically modified organisms, GMOs. That's where we produce a plant that's resistant to pests, or resistant to pests like insects, or it doesn't need as much water, and as a result, or it's resistant to fungal organisms that destroy crops. We can make these really resistant plants that are fast growing and giving us lots and lots of food very quickly. So this is an area, the reproductive cloning can definitely influence agriculture. Same thing if you do that with a cow that produces a ton of milk and is really healthy and resistant to disease. You clone that animal and make lots of them, then we've got really good agricultural and milk production, agricultural benefits of milk production. But what are the risks of that now? Yes? Oh yeah, methane. You know where methane comes from in cows? What? Very good word, yes. Flatulence. You know what flatulence is? Passing gas out the backside, right? When cows eat a lot of grass, and they have special enzymes to break down the cellulose, and in their gut that produces a lot of methane, and that actually contributes to global warming. The methane coming out the back of cows, because we have so many cows in certain factory farms. You know, they have thousands of cows out to pasture producing a lot of methane to the environment, which contributes to the greenhouse gases and global warming. So yeah, if we can produce cows that produce less methane, that's better for the environment, because we need to eat, right? We need to produce that milk for all the dairy products and burger and all that. So therapeutic cloning and reproductive cloning, the difference between them: therapeutic cloning, think of it as therapy. We're trying to fix a human disease by making some tissue that is functioning better than the person's tissue that maybe was damaged by a mutation or cancer or just genetic disease. So reproductive cloning is making a brand new individual, whether it be a plant or an animal. So this is just the process and how they do that. We're not going to go into the details of that, but this is reproductive cloning. This is therapeutic cloning. And where do they get the cells for therapeutic cloning? There's two different sources. One has a lot of debate behind it, a lot of ethics. The other one, nobody cares. So when you hear the word stem cells, where they're getting those cells is from people that undergo in vitro fertilization. Do you know what that is? Some of you do, some of you don't. That's where a couple can't have a child naturally, so what they do is they stimulate the female to produce a lot of eggs. So they give her special injections, get her to produce a lot of eggs, and then they harvest those eggs. They take the eggs from her body once they're ovulated, and they harvest, because women only produce 1 egg a month when they ovulate, and then they shed that with the menstrual period every month. But if they hyperstimulate the female, they can get lots of eggs in one cycle, so they give her medicines to do that. They harvest those eggs, then they have the male produce a semen sample, which is much less invasive. He provides the sample. They take the sperm, and they take healthy sperm, so they treat the semen to get the healthiest, best swimming sperm and healthiest sperm as far as the nucleus being intact and all that and looking good. And they put the sperm and the egg together in a petri dish. I mean, that's, I'm simplifying it, but outside the body. That's what in vitro. In vitro means outside the body is where fertilization is happening. So IVF, in vitro fertilization, is outside the body. So this happens in a petri dish. Sperm and egg come together. It forms a zygote, which is it's a one-celled little person. And then that cell divides and divides and divides by mitosis, right? And then once it has grown to a certain point, and I don't know how many days they wait, but once it's grown into a certain point, maybe it's an embryo even, a couple weeks, then they implant it in the female, implant it in the uterus, and then it attaches and grows. Some people implant just the sperm and the egg into the fallopian tube. That's called it's called gift. I forget what it stands for, but anyway, regardless, they implant that in the female. Well, what do they do with these other ones that have grown in the petri dish? What do they do with these early little people, little embryos? What do they do with those extras? Because they'll have maybe 8 or 9 that they're growing, and they pick the best 2 or 3 that have grown the best, and they implant that in the female. So if all 3 take, she's going to have triplets, right? Oftentimes they'll take, the couple says, I'll take 2. That way we have a better chance. If one doesn't make it, at least we'll have 1 child. But if both make it, we'll have 2. And there's a lot of couples that have twins because of successful in vitro techniques, and they have twins. So what do they do with the extras that are still in the petri dish? There might be 5 or 6 in there. What do they do? Dump them down the sink? Some people say no, those are little people, right? Those were the product of conception. Sperm and egg came together, they're growing, those are future people. Some people freeze them and use them for future pregnancies. So that's what some people do. Some people say you can have them and adopt them out. You can actually adopt an embryo, a female can. If they don't have a partner and they want to have a child, they can adopt an embryo. And they'll ship them this frozen embryo to their local fertility clinic. They'll go in and the doctor will implant that into the female, and she's going to have a normal pregnancy that's not her sperm or egg, but she's going to grow that child with her uterus and have a normal pregnancy and delivery. So that's an option. Another one is you can donate them for stem cell research. And stem cell research then is using it for those other purposes, for the therapeutic cloning. Now some people say that's wrong. Well, another source of stem cells is human skin, because we're always making new skin cells every month. We're shedding our skin, so there are cells in our skin that are stem cells, and we can use those for therapeutic cloning. So stem cell research kind of has 2 camps. There's those that are using the embryonic stem cells, and there's those that are using the skin cells. Another source of stem cells is in the umbilical cord. So they can use umbilical cord blood from new babies for stem cells. And some people, when they're treating cancer and they're doing bone marrow transplants for a child that has leukemia, they find that a good rich source of cells for replacing cancerous bone marrow and making new bone marrow is in the umbilical cord blood of a sibling. So some people have gotten pregnant and had a child for the sole purpose of getting that umbilical cord blood to help their child with cancer treatment. But they still, of course, have a child as a result of that too, but it's not uncommon. And some people will save you, like some of you that are going to have kids one day, they're going to ask you, they're going to give you a pamphlet, would you like to save your umbilical cord blood? And some people harvest it when their baby's born, and they keep it on ice and pay a monthly fee to have this umbilical cord blood available should something happen to their family. Now, who would do that? I would think if you have a history of childhood cancer in your family, yeah, it might be worth considering that. But they offer it to a lot of parents, and most of them say no because it's expensive and isn't necessary. Probably not. Okay, so that's a little bit about stem cells. So prokaryotic cell division. What is a prokaryote? Going back to the beginning of the semester, we talked about eukaryotes and prokaryotes. Bacteria, yeah, so I want you to write that down so you remember we're talking about bacteria when we're talking about prokaryotes. So bacteria don't have a nucleus, right? They don't have organelles. So how do they multiply? And we know they multiply, right? If you've ever had a strep infection, you start to feel a little sick and bam, you're down in bed and out and miserable with a bad sore throat, right? And pussy tonsils. We know that bacteria can divide very quickly. And they do it by a special process. Well, they have this DNA, which is we call nucleoid, which is the region where we find the DNA. We call that nucleoid. And they split in a process called binary fission. So DNA replication still occurs, and then the cell splits. So there's not a lot to divide, right? Because there's just the DNA, it replicates, and it splits. So these are exact copies. So they are identical to the original cell. It's asexual, which means it doesn't require another bacterial cell. So this is one way in which binary means two and fission means to split. So we have replicated chromosomes and those cells split. So we can see that the nucleoid is in the middle of the cell. It replicates. The cell wall, remember cell wall and cell membrane in bacteria, cell wall and plant cell, I can't talk. The plant cells and the bacterial cells have cell walls. But in bacterial cells, the cell wall does pinch inward. We don't call it a cleavage furrow, but the cell will elongate, pinch inward, and split, and you have two brand new cells. And when we look at how bacteria grow, it's called exponential growth. So if I drew a little picture here on the side, if this is a graph, and this is time on the bottom, and this is number of cells on the side, if I look at how bacteria grow, they go like this. And this type of growth curve is called exponential growth because we have rapid reproduction over a short period of time. So this is where you're just starting to feel sick, or maybe not even sick at all. Maybe you're just kind of hanging out and you have some bacteria that has entered in through some portal of entry, and then now you're really sick during this period of time, right? So exponential growth we see with binary fission because it's rapid. Each new cell is constantly undergoing this process of binary fission. So when you look at cancer, cancer is abnormal growth of cells. It's mitosis gone wild. If something is benign, so if we have tumors that grow that are not cancerous, we say it's benign. And you just have a lump in a certain spot. And women often have this in breast tissue. Breast tissue is sometimes lumpy and bumpy, and people get little fibrous cysts. And those are the result of hormones. So it's just with the cycle of hormones, sometimes it's pregnancy, sometimes it's just the menstrual period, people get little lumps and bumps. And a majority of tumors detected in a mammogram are not cancerous. But how do you think a woman feels when she gets that phone call after getting a mammogram saying, we want you to come back in, we found something? It's very stressful, right? But a majority of women, it's not cancerous. So that's important because maybe your parents are at that age. A lot of you in here are in your late teens, early 20s. Your parents might be in that age where they're getting those mammograms and getting positive, flags. Oftentimes it's fibrous. So it's not a reason to be upset yet, but it's concerning. The term malignancy means it's cancerous. And malignant means these cells grow and they enter into the lymphatic system, which is kind of our drainage system that we find in all of our body tissues. We have this excess little duct network that drains excess fluid from our tissues. These cells break off of these tumors and they enter into the lymphatic system. And the lymphatic system hooks up with the blood supply eventually, and now you've got cancerous cells in the blood supply, and then they invade other tissues through the bloodstream. So they kind of get on the highway of our bloodstream, and then they get off the exit ramps into different tissues. So that's why breast cancer often spreads to the brain and into the bones, because that's kind of the network of blood supply where it breaks off. So a person with a brain tumor, they'll go in and get a biopsy of that brain tumor, and they'll find that it's breast cells that are in the brain. So when we talk about cancer spreading, it starts in an area and then it spreads to other tissues, but it's those cells that were mutated and gone away from the tissue. So they invade nearby tissues and they can detach and lodge in different tissues. We call that metastasis. So metastasis, this is when we stage cancer. If you have a relative that calls up and says, I have cancer, and it's stage 4, that is a bad sign. That means that cancer started, well, let's say it's breast. It started in the breast, and it's spread throughout the body, and it's found in other tissues. It's found in the bone and the brain, and we do a special scan called the positron, what is it? Oh my gosh, I know what it's called. I'm forgetting it now. Positron emission tomography, PET. It's called a PET scan, and they look for active cells, cells that are actively dividing, light up on a PET scan, and that's where they look to see if somebody has cancer that's spread to different parts of the body. So metastasis means it's spread to all over the body. So the chance of recovery from stage 4 is way less than stage 1 and 2 cancers. So stage 1 cancer means they found a tumor in the breast and it's only in the breast, nowhere else. I have a relative that recently had that. So they removed the lump from her breast. They gave her a pill to prevent new ones from forming. She's got to take it for 5 years and she's clear. They don't want to see her again. She's fine. Stage 2 means they found a tumor in the breast or somewhere else, whatever tissue you're talking about. It's talking about breast cancer because it's so common. So stage 2 means they found a tissue in the breast and they found it in a lymph node near the breast. So that's stage 2A. Stage 2B, they found a tumor in the breast, they found it in a lymph node near the breast, and they found it in a lymph node on the other side. So it's spreading through the lymphatic, but it's not in the blood and it's not in the tissues. That's very treatable. So stage 1 and 2 cancers, very treatable with chemotherapy, surgery, radiation, you know, all the different techniques they use. But when you get into stage 3, that means it's spreading beyond and it's found in a distant site. Then it's a little more difficult. Stage 4, even more difficult. But do people recover from stage 4 cancers? We've got great chemotherapies out there and people have recovered from stage 4 cancer. So it's never a hopeless situation when we have stage 4 cancer, but it's much more serious and it requires aggressive treatment. And that's where some people decide, you know what, I don't want to do the treatment because it's so, when we kill cells, we're killing our own cells, aren't we? Because we said these tumors are our own brain cells dividing out of control. If it's brain cancer or breast cells, colon cancer, colon cells. So it's our own cells we're trying to kill. What happens when you give chemicals to kill the own cells? It's going to kill other cells as well. And it kills other cells that are rapidly growing in the body, which we need. Like your intestinal tract has cells that continue to grow. So people get really bad mouth sores. When I worked on pediatrics, we had a little boy, he was 5 years old, getting chemo for cancer, and he had open sores in his mouth because the chemo was killing the cells inside of his mouth. And all he wanted to do was suck on a washcloth all day long, cold washcloth, because it hurt his mouth so bad, this chemotherapy. It kills the immune system because those are rapidly dividing cells. So people, again, influenza, right? If we don't get our flu shots and we're in contact with people undergoing chemo, they're at risk. So when we look at classic chemo or cancer cells, they do not undergo apoptosis. They have mutations, but they continue to grow out of control. And they don't function correctly either. So if someone has cancer, they're going to continue to grow out of control. So we said they undergo metastasis. They also undergo angiogenesis. Make sure you know this term, forming new blood vessels. So here we have one little mutation in a cancer cell. It grows out of control. It invades the lymphatic and blood vessels, and then it spreads and forms a tumor somewhere else. So we have special genes that control the cell cycle. When they become mutated, they cause cancer. We call them oncogenes. And then again, we have tumor suppressor genes, and when they become inactive, that promotes cancer. So these two genes, oncogenes and tumor suppressor genes, when they're not functioning properly, that promotes cancer. Another thing is we have these telomeres at the end of our chromosomes. And if they get too long, that causes cancer.